Ага, хорошо. Спасибо большое. Вы запустили, да? Все, спасибо большое. До свидания. Guys, do you hear me? Guys, start coming. Guys, start coming for a lecture. Guys, do you hear me? Let me know that you hear me, guys. First Division, International Medical Faculty, third year, First Division. Start coming for lecture. Oh, now I can see that some students are coming already. Thank you very much for finding the lecture room and the link. Spread this link, please, if possible. So, guys, start coming. I supposed to start already. And today's topic is emerging and re-emerging viral infections. Guys, where are you? Guys, where are you? Start collecting, guys. Do you hear me? Guys, do you hear me very well? Guys, where are you? You, my dear Nur Sibhatullah, please send a link in IFM1 third year. Okay, guys, I start. Okay, so today our lecture will be devoted to the emerging and re-emerging viral infections. But of course, we won't talk about all of them. I would like to talk about most dangerous out of them that is recognized now as viruses that cause, okay, Garg is here, very well, as viruses that cause the so-called African hemorrhagic fevers. So, and you can see one of the very first electron photograph of those viruses. And you see by their shape that they are so-called filamentous viruses. They are very thin, small in width, but very long. And first, before we start discussing particular viruses that are recognized as emerging viruses, so we discuss the term that we use in our lecture, that is emerging and re-emerging diseases. So that is the definition by CDC. That is what kind of infections are recognized as emerging or re-emerging or diseases. These are new or re-emerging or drug-resistant infections whose incidence in humans has increased within the past 2 decades, or those incidents threatens to increase in the near future. So we have so many examples over the emerging and re-emerging diseases. And we have these examples in both bacteria and viruses. But of course, much more examples will be seen in viral infections and in viral diseases. And if you would like to give examples, these are Ebola and Marburg diseases, HIV infection with AIDS, this is the example, or Lassa, Hunin, Machupo, fevers, hantavirus, pulmonary diseases. Then you know all together SARS, MERS, and coronaviruses are now recognized as the new infections or emerging infections. So the infection caused by prions, that is bovine spongiform encephalopathy caused by prions, is also recognized as emerging diseases. So we have so many examples. But today we concentrate on the only one, that is Ebola and Marburg viruses from the same family. So, and if we are talking about the classification of this virus, so this is a group 5 according to the, according to the uh, uh, Western classification, the virus with -N single-stranded RNA genome. So, and the order is Mononegavirales and family Filoviridae that reflects the morphology of this virus, that is filamentous. And here we have genus Ebola virus, where we have two genera. Marburg virus, that consists of only one species, and Ebola virus. Not so far ago, so before we knew 4 of them, but several years ago, the new Ebola virus had been discovered, and altogether now we have 5. And these 5 you can see here, they are seen as Ebola virus Zaire, that is called ZBOV, Sudan Ebola virus, that is called CEBOV, Reston virus, that is called REBOV, Thai forest virus, that is called, because Ivory Coast, it had been found there, and it is called IceBOV. And the last one was Bundibugyo virus, BDBV. So those 5 viruses are Ebola viruses. And you can see here also the 5 identified subspecies of the Ebola virus. But they are not all equally pathogenic for humans, so that's why they are not equally important for the medicine. So, what is common for Ebola and Marburg viruses? They are both highly virulent. And both these viruses cause so-called African hemorrhagic fevers or filovirus fevers, my dears. Some other viruses also may cause hemorrhagic fevers, right? But to underline that this is caused by filoviruses, we may call it filovirus fever. So, as a result of infection, they are present in many organs of infected people and in many organs of infected animals. So, they have multiple ways of transmission. They can be transmitted by close contact with any body fluid, and they also can be transmitted by indirect contact. This is uh, transmission through the contaminated instruments. And because they are highly pathogenic and highly dangerous, the special caution must be used in handling needles and sharp instruments when we are dealing with this virus, because the injury with this may be really very dangerous. So, apart from their really dangerous natural pathogens, filoviruses are classified as category A biological agents. What does it mean? These agents are viruses, bacteria, or other germs that have a high potential for adverse public health impact and large scale dissemination. Apart from viruses, we study several bacterial pathogens that can belong to this group. Do you remember? These are anthrax, smallpox, pestis, uh, Clostridium botulinum, that causes Yersinia pestis, that causes plagues. Clostridium botulinum with botulinum toxin, one of the most potent toxin on the earth. But then also, apart from the bacterial infection, we have several viral pathogens that may belong to this group. And of course, out of all, the viral hemorrhagic fevers viruses or viruses that cause hemorrhagic fevers are recognized as most dangerous, and according to the um, safety precautions classification that should be undertaken to contain the infection. This is uh, type 4 or level 4, biosafety level 4 according to the USA classification, and biosafety level 1 according to the Russian Federation classification. So, and you can see here the special suit that's supposed to be worn because the filoviruses require maximum containment facilities for laboratory work. I would like to repeat biosafety level 4 regarding USA classification. So now we won't discuss any other emerging or re-emergent diseases and concentrate on the Ebola virus. And it is a filamentous virus, and you can see it here. It is, and so this is a typical snake-like structure. It is a enveloped virus, and the genome is negative strand RNA virus. So the virion appears as U-shaped, six-shaped. Sometimes it forms branching forms, and sometimes it forms snake-like structure. So it is very thin. So the width is 60 to 80 nanometers in diameter, but an average length of these particles can be about 1000 nanometers. But the length can be extremely variable, and this is 1000 nanometers is the average, but it can vary from 600 something to 14,000 nanometers. Can you imagine? Those particles are really large. Few words about the viral genome. So it is a one molecule, not fragmented, negative sense RNA, totaling 18, almost 19,010 nucleotides in length. So it is interesting that this genome codes for one non-structural glycoprotein that is called secretory glycoprotein. So that have the very special importance in the pathogenesis. And one more glycoprotein that form peplomers. So it means we may have two glycoproteins. The one that is secreted to outside, we call it secretory glycoprotein. And you can see here secretory glycoprotein, it is encoded by the viral genome. And the glycoprotein that will be not secreted but form peplomers. Apart from this, 6 more structural proteins are produced. And this is the nucleoprotein. Then the viral proteins 30, viral proteins 35, 40. Then the polymerase, viral polymerase protein is usually we call it L protein because it is very large. VP40 that acts as a matrix protein and VP24. So these are the proteins that are encoded by the viral genome. So inside the virion, we have a structure that is called nucleocapsid structure that is made up of nucleic acid or genome that we had discussed already. And this genome is encased in the nucleocapsid that is here. The nucleocapsid structure consists of a helically wound capsid with a diameter 40 to 50 nanometers. The capsid surrounds a central channel that is 20 to 30 nanometers in diameter. And the structural proteins associated with the nucleocapsid are no NP protein, you can see it here, then VP30, VP35, and of course, apart from those structural proteins, we have so-called L protein or polymerase. And you can see it is a really large protein that is fixed with the 5' end of the minus sense RNA. So then this nucleocapsid is covered by its so-called envelope. So that is lipid bilayer membrane taken from the cell membrane of the previous host, and it is derived from the cell membrane. And then the structural proteins associated with the envelope are VP40, that is a matrix protein that lines that is incorporated into the membrane, and 24 that makes the complex with VP40. So VP40, VP24. Then viral glycoproteins are inserted into the envelope, and there they form spikes with the about 7 nanometers in length and 10 nanometers apart from each other. Few words about the virus genome replication. The glycoprotein function, of course, is the attachment to the host cell membrane. And as soon as it binds, the virus enters by a receptor-mediated endocytosis, we think. And usually, as other minus sense RNA proteins, except for the orthomixoviruses. It replicates in the cytoplasm. The envelope usually fuses with the endosome, and the nucleocapsid will be released inside the cytoplasm. Because the minus sense RNA virus keeps within the L protein, that may start transcription, and then the L protein produces individual mRNAs for translation. These individual mRNAs code for the protein that will be later on translated. But then, apart from this, from minus sense RNA, viral genomic RNA, the the copies is here. The 4 is the transcription of minus sense RNA into full length RNA template. Because, my dears, these are not full length mRNAs. So the full length RNA or positive sense mRNA can be used as a template for transcription to generate many minus sense viral genomes. When individual mRNAs are translated into individual proteins, the development of the new nucleocapsids will be produced, because the assembly starts with the new genomes that will be coated with the proteins to produce viral cores. The newly synthesized glycoproteins can be of two types. And the glycoprotein that make paplomers will be taken to the cell membrane, and you can see here the red dots. These are the glycoproteins arranged into the paplomers, modifying the cell membrane. Then new nucleocapsids or new cores will be stacked up in the cell, forming crystal-like bricks. Can you see here? These are new nucleocapsids or cores that will be arranged like this in a brick-like structures. They will be aggregated. And in this aggregated manner, these batches can be taken towards the cell membrane. And the nucleocapsid then will undergo the so-called envelopment. So they will be wrapped in the cell membrane. So the rate of Ebola virus multiplication is very high. So, and the infected cell becomes packed with these crystal-like blocks very quickly of viral particles. You can see electron microphotograph of this. And the bricks form at center of the cell, and then they will be transported, grow outwards, and migrate to the cell membrane. On reaching the cell membrane, you can see how the bricks dissolve into hundreds of individual virions. And this huge amount of virions infiltrate the bloodstream, and it is believed that droplet of blood may contain up to 100 million virions. Can you imagine? And these are found here. You can see the bricks that are formed in the infected cell, and then what happens, how they are released. And here you can see it in higher magnification. So a few words about the pathogenesis of the disease. the uh, because the Ebola virus multiply very quickly, and the Ebola virus particles infiltrate into the bloodstream, then through the bloodstream, the virus can be spread to many organs. So, of course, uh, some cells may be infected with the Ebola virus, and they also may serve as the factor for spreading Ebola virus. And macrophages that are infected with the Ebola virus will act as a tissue factor, contribute to cascade that may cause disseminated intravascular coagulation. And also, the cytokine production may cause the activation of the dead cell receptors, cause apoptosis of the infected cells. And when other host cells or dendritic cells will be infected with Ebola virus, they may cause it may cause dysregulation of the co-stimulation of the immune cells to develop uh, good immunity against Ebola virus. So the virus replication causes direct damage of endothelial cells of blood vessels and extensive tissue necrosis in lymph nodes, spleen, liver, and lungs. So apart from the virus, the Ebola glycoproteins attribute to disruption of endothelial cells and the widespread hemorrhage followed by edema and hypovolemic shock that may be a cause of the death. So the Ebola virus is believed to spread without triggering of the immune response because surface glycoproteins binds neutrophils via Fc gamma receptor involved in neutrophil activation through interaction with the complement receptor type 3. And neutrophil activation is inhibited via preventing of CR3 binding to CD16B blockage by the viral secretory glycoprotein. The virus infects dendritic cells, important for the initiation of cellular immune response caused by NK cells, B cells, and T cells. So infected dendritic cells fail to mature. But if maturation doesn't occur, so the dendritic cells will not perform the activation of T naive cells because of lack of secretion and dysfunction of the co-stimulatory signals. And that is a very important reason why the uh, immunity is not arranged very well against the uh, Ebola virus. So that's partially how these viruses escape immune response by affecting dendritic cells and also by affecting uh, neutrophils and macrophages. There are some more ways how uh, uh, Ebola virus may escape or evade immune response. So, and that's very important that you might remember or you might know that when we are dealing with the RNA viruses, they're very important uh, mm, reagent that may influence and may downregulate the infection is interferon alpha and beta that have direct activity, and also interferon gamma that will be responsible for upregulation of cell mediated immune response. But the Ebola virus use its viral protein, that is VP24, that blocks interferon signaling traffic for all both two types of interferons, type 1 interferon alpha beta and type 2 interferon gamma. It blocked it by interacting with karyoferrin alpha 1, the nuclear signal receptor that is used for tyrosine phosphatase STAT1 transcriptional factor. This interaction, in general, you may answer, prevents synthesis of cellular antiviral proteins, and those proteins are mainly ribonucleases that may cause degradation of the viral genome. Apart from VP24, the viral VP35 blocks the transcriptional factor interferon regulatory factor IRF3, and it is important for the expression of interferon alpha and beta genes and synthesis on interferon alpha and beta. So, my dears, an interferon-induced antiviral state will not be really formed. So, no interferon alpha beta activity is performed. Then don't forget also that evasion of immunity also is performed by the production of viral non-structural secretory surface glycoprotein that acts as a decoy for neutralizing antibodies. And we've seen the same picture in hepatitis B virus. Do you remember? When HBs antigen had been produced in huge amount and secreted out of the cell, so it causes the decrease of the effectiveness of the neutralizing antibodies. We see the same effect here. And also, the viral envelope glycoprotein induces infectivity enhancing antibodies because as soon as it binds, as the antibodies bind with glycoproteins, take a look here. It may increase the uptake of the virus's bad monocyte macrophage cells lineage, and the viruses will replicate in the macrophages, and inside them can be spread within the rest of the body. So, Ebola virus causes so-called Ebola hemorrhagic fever, and the alternative names are Ebola hemorrhagic fever or just Ebola disease. It is a rare, severe, mostly fatal infectious disease occurring in humans and some other primates. And mainly it occurs in Africa, in several parts of the Africa, so that's why they are usually called hemorrhagic fever. And by now we have very few cases when it occurred outside the African continent. And that is very important because those cases had been connected with the WHO staff members that had been working in Africa within the previous outbreaks, and then had been taken. Those people within the incubation period had been uh, traveling to their own countries, but then it was contained there, for example, in America, in Texas. So, few words about the epidemiology of the Ebola virus. By now, it is endemic, considered to be endemic in Africa. And you can see here several regions. And mainly several countries there can be involved, but of course, the most affected country had been the Republic of Congo. Uh, we know now that this is an emergent disease, so we're supposed to identify the animals that may keep these viruses without causing any severe outbreaks in this animal. We think now that reservoir for the Ebola virus is again we try to blame bats, fruit bats. That is the most probable candidate to be a reservoir of the Ebola infection. And uh, the fruit bats, according to the recent discoveries, could maintain the virus in the tropic forest because these mammals experimentally infected with the Ebola virus did not die. It means they may act as a reservoir without causing any severe disease and maybe not causing even clinical symptoms in the natural reservoir hosts. And in Africa, people regularly eat these animals, so this could be the way of transmission. Or another way of natural way of transmission is when fruit bats uh, eat uh, very ready fruits and they drop the ground. So primates or sometimes human being can pick up this fruit and can be infected by eating the infected fruits. So it is very important, and you can see here a photograph of the rural hospital in Africa. So, and it is very important. Then the most of outbreaks, at least at the very beginning in 1972, starting with 1972, they have been uh, transmitted as the nosocomial infection within the hospitals because of the poor medical conditions in rural hospitals that you can see here. So, the poor medical conditions may be due to the reuse of contaminated syringes and needles. And it is known very well that first case in 1972 of Ebola virus can be had been confused with malaria disease because on initial stages of these two infections we have the common symptoms. So then the patient so-called zero, that was the first patient in the first pandemic, had been treated against malaria, and by the way, the infection from this patient zero had been spread within the rural hospital and then further through the population. So then, care of the patients without barrier nursing also may be seen in the rural hospitals without masks and gloves. For example, if you suspect malaria that is not spread like this. So, and also one more very important epidemiological sign of the spreading of this disease. Traditional burial preparations that are performed to be the corpses of uh, dead patients uh, consist of washing of the corpses and cleaning of the or cleansing of the guards. So that is performed by the family members. That is a very well recognized way of the person to of the transmission of the Ebola virus. So and if you have a Ebola hemorrhagic fever patient, the body fluids of patients from vomiting, diarrhea, and bleeding represent an extreme biohazard, and all these body fluids can contaminate a huge amount of Ebola virus. And also, this virus can be transmitted by indirect contact, for example, through shaking hand and through some fomites. So, if quite quickly it is touched by uh, individuals. So, not 72, 76, sorry. First, the Ebola virus had been discovered in 1976, and the first epidemic had occurred in the Zaire and Western Sudan in Africa. The mortality rate of these and the next epidemics was calculated as 50 to 80% of mortality, and that is really very high. The virus got its name after the Ebola River in the Democratic Republic of Congo, formerly Zaire, near the area where the agent had been found. So, within next years, the recognition of the Ebola virus species happened. So, in 1976, two first species of Ebola virus had been identified. Zaire Ebola virus with a case fatality rate up to 88%, and Sudan Ebola virus with a case for fatality rate much lower, but still very high of 53, around 53% mortality rate. So both these species had been demonstrated to cause human infections and spread fast from human to human. The third Ebola virus had been recognized in 1989, my dears. Take a look here. And it had been called Reston Ebola virus, or Rebo right now, and was first to isolate from the Philippines to the Hazleton Primate quarantine unit in Reston, Virginia, United States. But then, and there is a very good movie, by the way, that describes this appearance of the new Ebola virus. And it was the emergency situation because it had been found in animals. But then this virus appears to be spread only among monkeys. So it appears not to be so pathogenic for humans that even individuals infected with this virus didn't produce any clinically manifested diseases. The fourth type is called the Ivory Coast Ebola virus, or alternatively Thai Ebola virus, was identified in 1994 from a single human case in Cote d'Ivoire, and the species was named after Thai forest in the Ivory Coast, where the natural infection of chimpanzees occurred. And in 2003, 120 people had been infected and died in Etombi, Republic of Congo, which has been the site of four recent outbreaks, including one in 2005. And there are much more epidemics occurred since 205, and the main of them had been produced by Zebov and Sebov. Then finally, the Bundibugyo virus had been discovered, and it also could cause fatal infections in humans. So, the first recorded cases, or recorded case, or so-called patient zero, occurred in August 26th in 1976, It was a 44-year-old school teacher that just returned from a trip around northern Zaire from their family. The patient had a high fever, and initially his disease was recognized as possible malaria case. And inside the hospital, he was given a shot of quinine. You know that is anti-malaria drug, well recognized anti-malaria drug. And then visited the hospital every day. And because he had some contact with other people visiting the hospital, uh, he had been patient zero in the epidemic by infecting other people. So he had around four symptoms. He had uncontrolled vomiting and diarrhea, dizziness, headache, and trouble breathing appear in the week later after the initial fever symptoms. And then the bleeding began from his mouth, nose, and rectum. And the patient died 14 days after the onset of the symptoms. And this virus has caused more outbreaks than any other Ebola viruses. And in some epidemics, the mortality rate reached 90%. So here you can see the initial uh, uh, diagram or graph of the most deadly Ebola virus species. uh, regarding the first outbreaks. And you can see here that uh, the very little was the first one with the rate up to 83%, and then 88% in 1976, and even 100% in 1977, then a bit less, and finally 90% lethality in 2003. So, the second one, so-called CBOV Ebola virus, was the second species identified in 1976. And the fatality rate is much lower. It may vary from 50 to 60 something percent, and the average is recognized estimated as 54% of mortality. So in 2004, during the fourth outbreak of the disease occurred in Sudan, the case fatality rate was quite low, 25%, and not so many cases occurred. So regarding history of rest on Ebola virus, it had been isolated in 1989 from Cynomolgus macaques. This virus is highly lethal for monkeys and can be spread through air droplets. And it was shown that how it had been spread within the animal house in monkeys. In 1989, the infected monkeys were imported from Philippines to the Hazleton Primate Quarantine Unit in Western United States. And then the Western Ebola viruses, apart from this, had been discovered in 1992 in Italy, in 1996 in the Texas, USA, but none of the primate handles tested positive for this virus become ill. So then the next one, Ivory Coast Ebola virus. This species was named after the Thai forest in the Ivory Coast, where the natural infected chimpanzees were discovered. In 1994, the virus had been identified from a single human case in Cote d'Ivoire. It was a scientist that became sick and then recovered after conducting necroscopy on a chimpanzee. Ivory Coast Ebola virus can be passed to humans from infected chimpanzees. In 2003, 120 people died in Etambi, Republic of Congo. This area has been the site of 4 recent outbreaks of the uh, Ivory Coast Ebola virus. So, a few words about the uh, latest discovery of the or newest discovery of Bundibugyo Ebola virus. It is closely related to the Ebola virus. It causes very severe viral hemorrhagic fever in humans and is a so-called select agent, a potential that with the potential to pose a severe threat to public health and safety, according to the definition produced by CDC Center in the United States. Or it belongs to risk group 4 pathogen, requiring biosafety level 4 equivalent containment by WHO. Ebola virus diseases outbreak due to Bundibugyo infection occurs in Uganda, Bundibugyo district, and in Province Orientale, Democratic Republic of Congo. And you can see here the number of the human cases and the mortality rate. This virus was first described in 2008 as a single member of a suggested new species, Bundibugyo Ebola virus, which was suggested to be included into the genus Ebola virus. So, a few words about the Ebola virus infection. Incubation period is around 2 to 21 days. Symptoms are variable, but then the onset is very sudden, and initial symptoms usually include very high fever, more than 38 point something degrees Celsius, very severe headache, dizziness, sore throat, muscle pain, joint ache, fatigue, nausea, and abdominal pain. These symptoms or these manifestations progress to internal and external bleeding from any open in the body, even if the shot is given by syringe, then it starts bleeding from the site of needle injury. The secondary symptoms produced in African Ebola virus fever are severe vomiting, bloody diarrhea, red eyes from swollen blood vessels, and very severe hemorrhagic rash that originates from severe subcutaneous bleeding. Bleeding from the nose, mouth, rectum, genitals, and needle puncture sites is seen. Low blood pressure, less than 90 millimeter, fast and weak pulse, symptoms owing to the internal organ damage, the liver and kidney necrosis. And then it leads to hypovolemic shock. And usually it takes from 7 to 14 days from onset of the symptoms to death. So it's high lethality rate. Now the question is, how do we perform the diagnosis of this disease? So the growth of the virus is possible, but the specimen should be handled with extreme care. I would like to remind you this is a level 4 isolation procedure that are not routinely available in the clinical hospitals and, of course, in the rural hospitals. So that's why quite often the rapid diagnosis is performed, and this is a detection of viral antigens and serum and any body fluid. This is usually performed by ELISA, and mainly if liquid is taken, some fluid is taken, or it is performed by immunofluorescent assay if the tissues are taken, and usually it is direct assay. Then, because of the high amount of the viral particles and that they are quite large, so the diagnosis can be performed by electron microscopy over blood plasma, but of course it is not available everywhere. Isolation of the virus can be performed because the practically virus grows in a wide range of animal and human cells. So we can grow it in the various cell cultures. For example, the very regular Vera. So then the animal inoculation can be performed. Usually out of small animals, the guinea pigs can be used. Serology can demonstrate filovirus antibodies in serum of recovering patients, my dears. But serology is not really in use of the diagnosis of the acute disease because antibodies are usually developed when the very severe symptoms already starts. That can be important in clinical recognition of this disease plus the epidemiological information about the outbreak that occurred in the particular area. So it is not really used for diagnosis of acute infection, just to confirm the disease and see the result in serum of recovering patients. Then, of course, the reverse transcriptase polymerase chain reaction is performed, and it is used to detect the viral genome in any secretion where the facilities for reverse transcriptase PCR is possible. Sorry, this is RT, reverse transcriptase. So, few words about the treatment. Maintain, maintain, then to maintain the electrolyte balance and renal function. That is to renew the electrolyte balance. Then the treatment directed of combating the hemorrhage and shock. These are the examples of symptomatic treatment. Pooled plasma from seropositive convalescents and interferon therapy can be used as antiviral therapy and had been tried in a patient with filovirus infection. Right now, an experimental monoclonal antibodies against three different regions of glycoproteins prevent adhesion of the virus entry, so it also can be used in treatment. Then the treatment with pooled plasmas from seropositive convalescents and monoclonal antibodies is the specific treatment, my dears, right? So because those preparations are active against this particular virus only. Apart from that, experimental nucleoside analog inhibitors of S-adenosyl homocysteine hydrolase were shown to be capable of inhibiting at least Zaire Ebola virus infection. It is a cell-encoded enzyme that uh, the inhibition of it indirectly prevents methylation of 5' cap required for viral replication. So the viral mRNA production will be inhibited. So no mRNAs, no translation. Ebola virus infection prevention and control. You can see here the most modern the biosafety level 4 costume that's supposed to be worn when working with biosafety level 4 pathogen. Working with biosafety level 4 pathogen. Of course, for prevention and control of the infection, the infected patient should be quarantined. Hospital personnel isolate ill persons through so-called barrier techniques. Then containment animals should be sacrificed. Handling of the contaminated materials requires very stringent isolation procedure. Then, how about vaccination? The recombinant vesicular stomatitis virus, Zaire Ebola virus vaccine, is produced in the United States. And also, vector glycoprotein vaccine, that is called HPV-IV, is produced in the Russian Federation. So these are two different vaccines, and they had been produced in the United States and the Russian Federation. So a few words about the vaccine that is produced. The first vaccine to be approved in the United States and European Union in 2019 was so-called recombinant vesicular stomatitis virus, Zaire Ebola virus vaccine, also known as Ebola Zaire Vaccine Live or ERV-WEBO. It is a recombinant replication competent viral vector vaccine. So it means it consists of the live attenuated recombinant virus of the vesicular stomatitis virus, which had been genetically modified to express the main glycoprotein from the Zaire Ebola virus wild type. This vaccine for adults can prevent Ebola caused by Zaire Ebola virus. When using but then in 2019, I would like to tell you that first time then so-called the RIN vaccination strategy had been used with this recombinant vesicular stomatitis Ebola vaccine. What is it? Ring vaccination. That was immunization of those people who are likely to be infected. So because of the contact. So in practically all contact persons had been vaccinated. And the strategy of vaccination showed very high level of protection. But the problem is this vaccine had to be given twice. So and because of the difficulties of the so a lot of people there, for example, in Congo, in rural area, they are migrating. So it was a really problem of um, people to come back for vaccination to the same point of vaccination. That was the difficulties with the vaccination. The devastating Ebola virus epidemic in West Africa in 2013 and 2016 accelerated the process of several vaccines development because then it was recognized the very need for anti-Ebola virus vaccine. And the clinical trial starts including the replication competent vesicular stomatitis based vaccine expressing EBOV glycoprotein vaccine that underwent the clinical trials within 2016 and 2019. So, preclinical testing in animal models demonstrated the prophylactic and post-exposure efficiency of this ZEBO vaccine, identified the mechanism of protection, and suggested it was safe for human use. And you can see there uh, mm, the genes over the vesicular stomatitis virus, of the vesicular stomatitis, and which gene out of the Ebola virus had been taken for vesicular stomatitis. It had been found safe in livestock, and it was shown that it causes the immunity pre exposure and post-exposure immunity. It means the animals first had been exposed to the virus and then vaccinated. Mm, so then the durability of the immunity has been shown. What does it mean? It means antibody and cell-mediated immunity had been produced. Then heterologous challenge. It means that the immunity against one type of the virus, for example, Zaire, also partially prevent the Sudan infection. That is called heterologous challenge. And also, this vaccine had been shown to be safe in immunocompromised individuals. Then the trials in primates showed the time to immunity, then the post-exposure efficiency, the neurovirulence of the vaccine, safety in immunocompromised individuals, and effect of pre-existing immunity to vector. The clinical trial, the first to third stages of clinical trials occurred in North America, Europe, and Africa. Although some side effects of the vaccination were observed, for example, effect of pre-existing immunity to vector, these clinical trials showed that this vaccine was safe enough and immunogenic in humans. So when this vaccine had been proved, it started to be used in African population. First, it was used in 2018 outbreak in Democratic Republic of Congo in Equator province, and has since been used extensively within 2018-2020 in Kivu Ebola outbreak with over 90,000 people vaccinated. That is really breakthrough in the prevention of the Ebola virus infection. So how about the Russian Ebola vaccine? It was announced in 2016 that the intention to conduct the field trials of two Russian vaccines involving 2,000 people. These vaccines are from Gamaleya University Institute, Gam Evac Combi, and Gam Evac Leo. The vaccine consists of live attenuated recombinant vesicular stomatitis virus and adenovirus serotype 5, expressing Ebola envelope glycoprotein of Zaire Ebola virus. The vaccines are targeted against the Makona variant of Ebola that was circulating in West Africa during 2013-2016 outbreak. Gam Evac Combi is a heterologous vesicular stomatitis virus and adenovirus 5 vector Ebola vaccine. There is also a version called Gam Evac with this homologous adenovirus 5 vector vaccine. As of 2015, Gam Evac Combi vaccine has been licensed in Russian Federation for emergency use. The Guinea government authorized the commencement of the trials on the August 9, 2017, and as of October 2019, Russia licensed the vaccine by local regulatory authorities and was reportedly ready to ship vaccine to Africa. My dears, you can see here that this adenovirus vector had been intensively studied starting from 2016, and this vector had been used as a vector for vaccine development against coronavirus. That's the technology and the strategy of this vaccine development had been discovered not so far ago, but then, fortunately, it was ready by the moment we needed against coronavirus, so it had been used. So, prevention and control, non-specific prevention and control. So, hospital personnel wear gloves, disposable face masks, gowns, goggles when caring for patients. The visitors to this area are strictly restricted. Disposable materials are removed and burned after use. All reusable materials are sterilized before use. All hard surfaces are cleaned with the sanitizing solutions. And proper disposal of waste and corpses uh, is very important. So, and the use safe burial practices are very important because at the very beginning of the outbreaks, starting from 1976, the doctors and nurses, according to the or from the WHO, had been sent to Africa, but the only thing they could do is to prevent the spreading of the virus through the unsafe burial procedure. It was really very important that time, but it is really important now. So, my dears, with this, I would like to finish my lecture, and uh, I think the next lecture will be delivered after we come from the Christmas holidays after 10th of this January. And I think the last lecture, if we manage, that will be about oncogenic viruses. My dears, I would like to remind the students that some teachers um, uh, arrange the reworking starting from the 27th of December. Within the week, you will not have any classes. So please take a look at their timetables and try to rework as many or to pass as many CPs as possible. With this, I would like to finish and see you in classes and on reworkings. Goodbye, my dears.